let's continue further. Let's talk about the gram stain that we were talking about. So gram staining, if I talk about, so the first thing, these are the headings under which I'm going to talk about. And gram stain is one of the differential stain. Already we all know what is like differential stains kya hote hain, and one of the stain is gram stain. Starting under these headings, I'm going to describe your gram stain, starting with the very first one that is called as the introduction of this thing. Let's jump towards the introduction wala part. Now under introduction, I would be mainly talking about who discovered in your gram staining. So gram stain was developed by Hans Christian Gram in the year 1884. And why do we perform a gram stain? Definitely to differentiate between the gram positive and the gram negative organisms. Done? Introduction is done. Second important segment we have to cover up under this part is nothing but the introduction ho gaya hai. Let's talk procedure. First then I will be shifting towards the principle. Let's start with something called as the procedure. Now under procedure, देखो gram stain if I talk about your Zn stain or acid fast stain, दोनों जगह पे जो procedure होता है, it is more or the less involving the similar steps only. How do I remember the procedure? First of all, remember the steps. Okay, there are total 4 steps. ठीक है? Remember the name of each and every step. First of all, there is something called as a primary stain. Second, we're going to apply the mordant. Third, we're going to do the decolorization, and lastly, we'll be applying the counter stain. ठीक है? तो पहले तो 4 steps clear हो गए हम लोग को. प्राइमरी स्टेनिंग के अंदर क्या होता था. फर्स्ट आई विल एक्सप्लेन यू दिस एंटायर डायग्राम एंड देन डिटेलिंग्स में मैं एक्सप्लेन करूंगा. आई विल एक्सप्लेन इन डिटेल व्हाट इज देयर एंड व्हाट इज नॉट इन हियर. ठीक है. तो फर्स्ट ऑफ ऑल इफ आई लुक ओवर हियर, देखो बैक्टीरिया लुक ओवर हियर, सारे बैक्टीरिया कैसे कलरलेस है. आई ऑलरेडी सेड यू इन द स्टार्ट इटसेल्फ कि सारे के सारे जो बैक्टीरिया होते हैं कैसे होते हैं, कलरलेस होते हैं. तो फर्स्ट जो हम प्राइमरी स्टेन अप्लाई करते हैं, all the bacteria irrespective कि वो ग्राम पॉजिटिव हो या ग्राम नेगेटिव हो, all of them will take the crystal violet stain, that is the primary stain, and each and every bacteria after the first step they will be appearing kind of violet to blue in color. ठीक है. Second step, we are going to add on the mordant over here. Grams iodine is okay used as one of the mordants and we apply it for 1 minute nearly, and definitely after each step you have to rinse it with the water in gram stain. ठीक है ना. So after doing also again all the bacteria have the primary stain intact in itself. What is the importance of mordant? In the next slide I will be explaining you what is the importance of this mordant, but for the time being just remember initially it was colorless. After applying the primary stain and the mordant itself, दोनों apply करने के बाद they appear kind of bluish purple. Okay, kind of आपके ऐसे purplish kind of appear होते हैं ये. Up step 3 is one of the most important steps, that is the most crucial step you should say, that is decolorization. Agents used are your acetone can be used, ethyl alcohol can be used, any of this thing, and only for 2 to 3 seconds, 20 to 30 seconds depending आप कौन सा agent use कर रहे हो, आपका duration change होने वाला है. ठीक है. After doing, can you see that these bacteria that I'm highlighting over here, these particular bacteria, they have lost something called they loses the primary stain. इनके अंदर से primary stain क्या हो गया है, गायब हो गया है, and now they have become colorless. Why so? These bacteria which on the application of a decolorizer, okay, they loses the primary stain and become colorless once again, those are your all type of your gram negative bacilli. All your gram negative bacteria will lose the primary stain on application of a decolorizer. Clear? Then आता है step 4 जिसमें हम लोग basically counter stain हम लोग add करते हैं. Counter stain के examples are 2, that is carbol fuchsin or saffronin. Again you have to apply it for 30 seconds and lastly you have to rinse it and you have to blot it dry. ठीक है. Now what happened? All your gram negative bacteria take up the counter stain, and the one which is going to retain the primary stain and appear purple in color, those are all the one which retains the primary stain are gram positive, which takes up the counter stain, they are a gram negative. So this is like a basic overview that I wanted to highlight under this segment. Clear है यहाँ पर. अब आते हैं detailings of each and every वाले segment में. So first of all, how do I remember each and every step? First of all, remember the name of the step. We know there are 4 steps. First step is you are going to apply the primary stain. First step, you're going to apply the primary stain. Second step, you're going to apply the mordant. Third step, you're going to do the decolorization. And at the end, we're going to apply the counter stain. After remembering all the important, what you can say, the names of the step, let's talk about which agent are used under each and every step. जैसे कि primary stain. If I talk about primary stain, all the para-rosa aniline dyes. कौन से dyes use हो रहे हैं? Para-rosa aniline dyes are used. And examples क्या है पता है? All the violet stains, जिसमें भी violet आ रहा है, those are used over here. All the violet stains. ठीक है. Gentian violet हो गया, methyl violet हो गया, और crystal violet for say. These are the important examples of your para-rosa aniline dyes. Any problem in remembering this segment? Let's talk about mordant. Mordant के नाम पे भी आज जो हम चीज यूज कर रहे हैं, so under the name of mordant, the thing that we are using is nothing but a grams iodine. What are we using? We are using grams iodine. ठीक है. So first primary stain के अंदर हम लोगों ने कौन से all the crystal violet, gentian violet, methyl violet जैसे para-rosa aniline dyes use कर रहे हैं. Mordant के लिए we are using grams iodine. Decolorization, there are 3 agents that we can be used as a decolorizer and those are nothing but your acetone can be used. Okay, so three agents can be used over here, and those are your acetone can be used, ethyl alcohol can be used, or acetone alcohol can be used. Any of these agent can be used. And lastly, counter staining purpose के point of view से safranin can be used or diluted carbol fuchsin can be used. Okay, so safranin or dilute carbol fuchsin, any of this stain can be used. So first सारे steps के नाम याद किए. Second, which agents are used, we all remembered all that part. Then comes into picture the duration for which you have to apply that particular agent. तो प्राइमरी स्टेन यूजुअली हम लोग 1 मिनट के लिए अप्लाई करते हैं. ठीक है. Mordant जो होता है that is grams iodine, it is applied for 1 minute. What is grams iodine? It is nothing but a dilute solution of iodine. Diluted solution of iodine is called as your grams iodine and it is applied for 1 minute. ठीक है. Decolorization के लिए we are using acetone. If you are using acetone as a decolorizer, it is applied for nearly 1 to 2 seconds. Okay, it is applied nearly for 1 to 2 seconds के लिए हम लोग उसको अप्लाई करते हैं. For 1 to 2 seconds we are going to apply. If you are using ethyl alcohol, then 20 to 30 seconds can be applied, and acetone alcohol use हो रहा तो nearly 10 seconds के लिए we can apply. And lastly, counter stain जैसे कि safranin या carbol fuchsin जो होता है, ये have to apply it for 30 seconds. I hope each and everything is very much clear till here to every one of us. Done with the steps, done with the agents, done with the duration. Lastly, we have to just explain that after doing the first step, what is the end result. Okay, so let me highlight it with the red color. Okay, so if you have a look after doing with the putting the primary stain, irrespective, देखो crystal violet is going to stain all the bacteria violet in color. So all the bacteria will appear violet in color, all the bacteria, irrespective of the fact whether it is gram positive or gram negative, फर्क नहीं पड़ता. What will happen? Each and every bacteria present in the slide would be appearing violet in color. That is the main thing. The first one. Second step में जब आप mordant add करते हो for 1 minute, after doing that, what is the main thing? What is the main thing? What's the role of this mordant? That is, it is going to form bigger diiodine complexes within the cytoplasm. Now, what is the role of this thing? Okay, it is going to bind to the dye. Iodine will bind to the dye, and dye के साथ bind करने के बाद bigger diiodine complexes would be formed, and in the cytoplasm would be formed. And this is very important for the return of the stain. The primary stain gets retained within the cytoplasm of the cell, so that okay does not gets off from that thing. Then comes your step 3. Step 3 is a decolorization. Under decolorization, it is going to remove the primary stain from a gram negative bacteria. What it is doing? It is mainly doing something that is called as, it will remove. What it is going to remove? The primary stain. And it's a decolorizer, na? It will remove the color. It is going to remove the primary stain from the gram negative bacteria. While if I compare it with the gram positive bacteria, it is going to retain the primary stain. Do you get why this thing is possible? Why is this thing happening? There is a question mark over here. The answer I would be uncovering under something called as the principle, जहाँ पे I will be talking about that why gram positive bacteria are going to retain the primary stain and the gram negative on the other hand is going to lose the primary stain on application of a decolorizer. I will be discussing it shortly under the topic called as a principle. ठीक है? Let's talk about counter stain. Counter stain के बाद जब मैं करूँगा तो secondary stains, सैफ्रनिन हो गया या डाइल्यूट कार्बोल फ्यूजन हो गया, 30 सेकंड जब अप्लाई करोगे, इट इज गोइंग टू इम्पार्ट ऑफ पिंक और रेड कलर. ठीक है, पिंक या तो रेड कलर हो जाएगा. टू विच बैक्टीरिया? डेफिनेटली द ग्राम नेगेटिव बैक्टीरिया. Okay, this wraps up with the segment. At the end of the entire procedure, you have to blot the slide dry and you have to examine it under the oil immersion objective. ठीक है ना? After rinsing and kind of drying the slide. After rinsing and drying the slide, you have to observe it under the oil immersion. I hope this sounds a bit clear to every one of us. Now there is there is an important point that I want to add on to this segment, and that is over here. See, under the counter stain, there is another example of a counter stain. Apart from saffron and dilute carbol fuchsin, there is another example, and that is called as a neutral red. And it is mainly used as a counter stain, for gonococci. ठीक है ना? तो neutral red gonococci के लिए use होता है. This thing we can add on. अब second important thing that I wanted to add on extra from this is, what is this this highlighted box, this note point over here representing. So just for an example, decolorization से related एक extra point मुझे बताना था यहाँ पर. तो decolorization से related, so let us talk about something called as your, let's take an example of ethyl alcohol, ठीक है, as a decolorizer. So there is a time limit over here, right? I am saying nearly 20 to 30 seconds you have to apply this thing, right? Now there are two possibilities, ठीक है, there are two possibilities. What if I say, if you allow the decolorizer for a longer duration, for some extra time, or you apply the decolorizer and you immediately wash the side, ठीक है, that is you allowed the duration, you reduce the duration of that particular sign. So this is the optimum duration, 20 to 30 seconds for ethyl alcohol. But what happened? you let the decolorizer stay on the slide for a longer duration, or you reduce the duration by immediately rinsing it off. So there are two possibilities. If it stays for a longer duration, it will be something called as your over decolorization. And if there is a feel like for a shorter duration, if the duration is reduced, then definitely there is something called as your under decolorization. What is the outcome? If it gets over decolorized, all the gram positive bacteria will also start losing the color. ठीक है ना? तो अगर ओवर डिकलराइजेशन हो रहा है तो व्हाट विल हैपन, द ग्राम पॉजिटिव विल आल्सो लूज द कलर, एंड अंडर डिकलराइजेशन मींस व्हाट हैपन दैट द ग्राम नेगेटिव बैक्टीरिया दैट यू हैव बेसिकली, ये स्टेप डिकलराइजेशन के वक्त ग्राम नेगेटिव शुड लूज द स्टेन ना, बट इफ यू आर अलाउइंग ओके द decolorizer to stay for a very short duration, what will happen, the gram negative won't lose the primary stain so properly. So that is, do not lose the color of the primary stain properly, and therefore again false result will come. I hope the complete procedure is very much clear till here. Let's shift towards the next important segment. So we are done with something called as the introduction. We are done with the procedure. Let's head towards the principle now. ठीक है, अब प्रिंसिपल की बात करते हैं. प्रिंसिपल में जरा थ्री इंपॉर्टेंट थ्योरीज, दैट इज वी हैव द पीएच थ्योरी, वी हैव द सेल वॉल थ्योरी एंड वी हैव द मैग्नीशियम राइबोन्यूक्लियर थ्योरी. तीन इंपॉर्टेंट थ्योरीज हैं, ऑफ विच दिस वन, that is your cell wall theory, is one of the most important theories or postulates that explains the basis of your gram staining. Let's describe each one of them in detail over here. Starting with the very first one, that is the pH theory. pH theory simply ये बोलता है कि ग्राम पॉजिटिव बैक्टीरिया का साइटोप्लाज्म जो होता है वो एसिडिक है, राइट, एंड जो हम लोग प्राइमरी स्टेन यूज करते हैं इट इज बेसिकली बेसिक इन नेचर. ऑब्वियसली बात है ना, एसिड एंड बेस, proper strong retention will take place. This is one of the major factors contributing towards the retention of the primary stain in a gram positive bacteria. कि भाई ग्राम पॉजिटिव बैक्टीरिया का साइटोप्लाज्म एसिडिक है, एंड जो हम लोग डाई यूज करते हैं प्राइमरी वाला है ना, इट इज बेसिकली basic in nature. Therefore, they both are going to be well, and retention would be there, and it is going to retain very well. Second, we add iodine as a mordant. Now, that also okay adds on to something called as the retention of primary stain in case of gram positive, because it forms the diiodine complexes, and in diiodine complexes, the size will increase, and the permeability of the cell wall of a gram positive will be reduced, right? And it's like the diiodine complexes are bigger in size, and they cannot cross the cell wall of a gram positive bacteria because peptidoglycan layers are multiple over there, right? And it is thin. So it is thicker in case of gram positive. Let's shift towards the next important one, that is called as the cell wall theory. Before going to the cell wall, I just wanted to add on, put some light on the magnesium ribonucleate first. Magnesium ribonucleate theory is nothing but, ये theory ऐसी बोलती है, this particular moiety, this magnesium ribonucleate moiety is present in gram positive organism में present होती है, and gram negative organisms में absent होती है. And if it is present in the gram positive, it is going to help retain the primary dye. Okay, it helps in the retention of the primary dye. That's it, the magnesium ribonucleate theory ends. One thing to just highlight over here, it is present in the cell membrane. pH theory जो होती है, it is mainly depending upon the pH of the cytoplasm, and ये cell membrane के ऊपर होती है. And जो cell wall theory होती है, it is mainly upon the peptidoglycan layer. यही gram positive bacteria, it is having thicker peptidoglycan, and gram negative one has a thinner peptidoglycan layer. So that is the same thing, cell wall theory explained under this particular segment. Let me highlight two possibilities. Either it can be a gram positive cell wall हो सकता है, या तो there can be a gram negative cell wall. ठीक है, gram positive cell wall की बात करूंगा तो it is having thicker peptidoglycan, tight cross linking, whereas here they have somewhere they would be having thinner peptidoglycan layer, and also the cross linking is not tight. Okay, they do not have a tighter cross linking, whereas here they have a tight cross linking and a thicker peptidoglycan layer. Nearly 50 to 100 layers thick होता है gram positive में. This is the main difference between the two. And since the peptidoglycan layer is more thicker in case of gram positive, they are going to retain the primary stain. And gram negative में thinner peptidoglycan, they will lose the primary stain on addition of a decolorizer. Additional point regarding the gram positive के साथ. Okay, so by talking about your gram positive bacteria, ग्राम नेगेटिव बैक्टीरिया के अंदर एक एडिशनल पॉइंट मुझे ऐड करना है, एंड दैट इज द प्रेजेंस ऑफ लाइपोपॉलीसैकेराइड लेयर. दिस लिपोपॉलीसैकेराइड लेयर जो अगेन सेल वॉल का कंपोनेंट होता है ना, ग्राम नेगेटिव बैक्टीरिया के, इट गेट्स डिसरप्टेड. ठीक है, दिस लिपोपॉलीसैकेराइड लेयर इज डिसरप्टेड ऑन एडिशन ऑफ अ डीकलराइजर. इट गेट्स डिसरप्टेड एंड देयरबाय इट विल प्रोड्यूस समथिंग कॉल्ड एज अ लार्जर पोर्स, है ना. जैसे डीकलराइजर ऐड होगा लाइपोपॉलीसैकेराइड लेयर्स के अंदर, लार्जर पोर्स विल बी फॉर्म्ड. ओके, दीस लार्जर पोर्स विल बी फॉर्म्ड एंड दे विल अलाउ दोस डायोडीन कॉम्प्लेक्सेस टू escape from the cytoplasm. Okay, they will escape from the cytoplasm. So this explains the entire cell wall theory as well. So far हमने क्या क्या cover up कर लिया है. So we are done with something that is called as the introduction is done, principle is done, procedure I have explained you. Now let's talk about the interpretation, then uses, modification and some examples I will be talking about. ठीक है? Let's talk about the interpretation over here. अब see, there are two possibilities again. Either the particular organism can be gram positive or it can be gram negative. अगर मैं gram positive की बात करूंगा तो कैसा appearance होगा? Violet in color होगा, है ना? Why it is violet in color? Because see, gram positive organism है ना, it is going to resist decolorization and thereby it will retain the primary stain and it appears violet in color. Done? Second over here, can you appreciate gram negative one? Gram negative bacteria are the one which are going to get decolorized. If they are decolorized, so they will take up the primary stain, they will take up the counter stain, they will lose the primary stain and they will take up the counter stain. And if they are taking the counter stain ले रहे हैं, तो they will be appearing kind of pink in color होगा. Yes or no? Look at the diagram over here. Can you appreciate the image over here shown? See, I'm highlighting here. See, multiple blue colored, purple colored, violet colored की gram positive bacilli can be appreciated in this entire image. And पीछे we can also appreciate the gram negative bacilli. Over here some gram negative foci can also be appreciated, right? So this is how we can see the interpretation, gram positive and gram negative under the microscope. This is how they are going to look like. Done and dusted with this part. Let's jump towards the next important segment. And so done with the introduction, the principle, the procedure, the interpretation, the uses are the next important segment we're to discuss, and at the end modification. ठीक है, so uses की बात करूं, what is the main use of a gram stain? Definitely to differentiate between a gram positive and gram negative organisms के बीच में differentiate करने के लिए we are using your gram stain, right? Why do we want to differentiate this thing? And on that basis there are multiple uses. On that basis there are multiple uses we can appreciate. See, first of all we want to differentiate this bacteria. Identification purposes के लिए use कर सकते हो. To start an empirical treatment, see if you perform something called as a gram staining, you got to know that like the organism is gram positive or gram negative, है ना? एक preliminary clue मिल जाता है, so that you can start the basic treatment ना. That हाँ, if there is a gram positive bacteria, so and so it can be expected as per the symptoms and stuff. So we can start the prescription, है ना? So we can start the treatment of that particular patient. For identifying some fastidious organisms like the Haemophilus, right? Some anaerobic organisms like Clostridia. Then yeast, very important guys. Candida, Streptococcus. These are yeast. These are not bacteria, but still gram stain is used for them and they appear gram positive. They will be appearing violet in color under the microscope if you do something called as a gram stain on Candida and Streptococcus जैसे yeast organisms. And lastly, to check the quality of the specimen, sputum specimen के examination के लिए we can utilize your gram stain. So these are the multiple uses. Just read it on your once and you are good to go with this segment. Let's jump towards the next important segment. And I guess there is modification left. Last thing, modification to cover up. So there are two modifications you should know. There are only two modifications that I wanted to highlight. See, first of all, there is something called as the Jensen's modification. And Jensen's modification is mainly for your meningococci and gonococci. Means all the Neisseria, है ना? All the Neisseria species के लिए we are applying your Jensen's modification of gram stain. And Brown and Bren modification is for the actinomycetes. So see, there are multiple modifications. Just for the example point of view, I am taking two important examples, and that is your Jensen's modification, and then there is your Brown and Bren modification. Jensen's modification is for the Neisseria, that is meningococci and gonococci, and Brown and Bren वाली जो होती है, it is mainly for the actinomycetes. I hope you would be remembering this thing. Then comes into picture the next important segment. With this, we wrap up with something called as a gram stain. One thing left, that is called as your examples over here. So examples, you're going to divide into two main categories. That is, first of all, I'm going to describe all your foci, and then I will be coming to the bacilli. Okay? Overall foci can be of two types. Either it can be gram positive or it can be gram negative. Right? If I go with the gram positive foci, there are two examples: Staphylococcus, Streptococcus. And if you go with the gram negative foci, two important examples: Neisseria and Moraxella. Then comes your bacilli. Right? So bacilli के अंदर we have again two possibilities. Either it can be a gram positive bacilli or it can be gram negative bacilli. See, for foci there are only 4 foci that you have to remember overall, out of which 2 is gram positive and 2 is gram negative. Remember this thing. And gram positive bacilli, that is, there is a way to remember it. There is A, B, C, D and L. Right? A stands for actinomycetes, B stands for bacillus, C stands for all your clostridias, right? Clostridium tetani, Clostridium botulinum, Clostridium perfringens, and difficile. All these things you can add up under this thing. And D stands for Corynebacterium diphtheri, Okay, it is your Corynebacterium diphtheria. I know it is starting with a C, but remember it with a D only, right? For the pneumonia. Okay, for the mnemonic, we can remember this thing. And lastly, we have your Listeria. L stands for Listeria monocytogenesum. Now, we can also add on to the gram-positive bacilli, that is your Mycobacterium TB can also be added to the list over here. Okay, that is also kind of gram-positive. But if I talk about Mycobacterium, the main strain is the next one, that is your Zn strain that we usually perform for it. Now comes gram-negative bacilli. Okay, see, one thing I want to say over here. Remember all the gram-positive bacilli. They are hardly 5 to 6 in a number to remember. Rest all of the bacilli would be gram-negative in nature. Rest all would be gram-negative. Just for example, E. coli, Klebsiella, Proteus, and all the bacteria other than this particular gram-positive bacilli को छोड़ करके बाकी सारे जो हो गए, they would be gram-negative bacilli. I hope this sounds a bit clear to every one of us, and this wraps up with the discussion of your gram-negative bacilli as well. So with this, we mark the end of something that is called as your gram stain khatam hota hai, guys. I have discussed each and everything, right? Let me just give you a quick recap. Introduction, who was the discoverer? We know it is Hans Christian Gram. Second, we so done with this thing. Second, we all talked about the procedure, guys, right? All the 4 important steps of procedure: primary stain, mordant, then decolorization, and lastly we all did the counter stain. And then principle, again there are 3 important theories to explain: the cell wall theory, the pH theory, and the magnesium ribonucleate theory. Then we all talked about the interpretation. Either it can be something called as a gram positive हो सकता है, या तो it can be gram negative हो सकता है. Done. And lastly we all also did something called as the uses. Uses की जब मैं बात करूंगा तो multiple uses we all had a look. And lastly there were 2 modifications of this thing that we all remembered. That is nothing but the Jensen's, and then we have the Brown and Bren modification. And at the end we also covered up a lot of examples about the gram positive and the gram negative foci and bacilli. This wraps up or sums up something called as all about your gram stain.
Thanks for watching. If you all found this video useful, then do like, share, and subscribe to my channel, which is AMSMBBS Lectures.